Welcome to where we dive deep into the marvels of science and technology. Today, we're unraveling the mysteries of PCR, the polymerase chain reaction. This technique is a cornerstone in the world of molecular biology, and it's as cool as it sounds. PCR stands for polymerase chain reaction, a method used to amplify a specific segment of DNA. Now, why is PCR important, you ask? Well, it's a game changer in disease diagnostics, enabling us to detect genetic abnormalities and infectious diseases swiftly and accurately. But how does it work? Simply put, PCR involves heating and cooling DNA in cycles, resulting in the amplification of the target DNA segment. So, in essence, PCR is like a Xerox machine for DNA. Imagine being able to copy and amplify a tiny, almost invisible segment of DNA into millions of copies. That's PCR for you. It's like hitting the jackpot in the lottery of genetics. At its core, PCR is all about amplification. But how does it work? Well, there are three key stages. First up, we have denaturation. This is where the DNA's double-stranded helix is heated to a high temperature, causing the two strands to separate. It's like unzipping your jacket. Each side now independent and ready for what's next. The second stage is called annealing. During this phase, short DNA sequences known as primers attach themselves to the start and end points of the target DNA. You can think of these primers as bookends, marking where our DNA copy needs to begin and end. Finally, we reach the extension phase. Here, an enzyme named Tuck polymerase walks along the DNA strand, adding nucleotides to form a new strand. It's like a diligent builder putting together the bricks to construct a new building, and voila, we've doubled our DNA. But PCR isn't a one-size-fits-all. We've got variations like RT-PCR, used for converting RNA into DNA, to quantitative PCR, which tells us how much DNA we've got. And let's not forget about digital PCR, breaking down DNA samples into smaller parts for ultra-precise analysis. Each type has its unique role, making PCR a versatile tool in the lab. PCR's real magic, though, lies in its applications. It's a detective, a researcher, and a problem solver all rolled into one. By amplifying DNA, PCR can identify genetic disorders, detect infectious diseases like COVID-19, and even help develop treatments. It's about not only understanding what's making us sick, but also about prevention, research, and paving the way for medical breakthroughs. Without PCR, the landscape of medicine and research as we know it would be vastly different. It's a tool that's enabled breakthroughs across the board, from forensic science to conservation biology. So, there you have it, the incredible world of PCR, a technique that's become the backbone of modern molecular biology. It's a marvel, amplifying the invisible to visible, transforming diagnostics and research. Thanks for tuning in to dive deeper into the wonders of science with us, and don't forget to like, share, and subscribe for more fascinating content. Until next time, keep questioning and exploring the world around us.